How complex can a designer microbiological process be? The early years of research in my lab were focused on this question. It is clear that there are many examples of connecting up sensors and actuators to achieve new function, but how far does this go? In this study, we pushed this style of engineering to its limits in the construction of a payload delivery device. The function of the payload delivery device is to enable E. coli to efficiently invade a cancer cell, sense their encapsulation within a phagocytic vacuole, and activate expression of a delivery program. In response to activation, the bacteria lyse themselves and lyse the vesicle, releasing a protein payload to the cell cytoplasm. Simple routes have been devised that require the acidification of the vesicle by fusion with lysosomes, and these conditions can denature the payload. Through engineering, the payload can be delivered under native conditions at much higher efficiency. At the high level, you can separate this device into distinct invasion, delivery, and payload processes. In a previous chapter, we discussed the engineering of the invasion process by transcriptional control of the protein invasin. We saw how we could restrict invasion to environmental niches. The payload device is itself a sensor-actuator complex device that is triggered by the chemical microenvironment of the phagocytic vacuole. In response, a self-lysis device and vacuole lysis device are activated, and the payload is released to the cytoplasm. To build this system, we constructed individual devices to build and test each component. We could monitor the activity of vacuole devices by fusing them to GFP, and we could watch the self-lysis device by controlling it with PBAD. In principle, we could also isolate the vacuole lysis device with PBAD, but there was no microbiological assay to watch that phenotype, and thus this component had to be analyzed in the context of the complete system. The self-lysis device was composed of 3 genes from either T4 or lambda-phage, which encode lysozyme, holin, and antiholin. Lysozyme, when accessible to the periplasm, will degrade the peptidoglycan, resulting in lysis of the cells. However, lysozyme does not contain a periplasmic localization tag. Instead, it gets to the periplasm by leakage through pores caused by holin. Holin is a short peptide that spontaneously inserts into the inner membrane, forming holes. The third protein, antiholin, binds to holin in the cytoplasm and prevents it from entering the membrane. Thus, this system is topologically similar to the threshold-gated circuit. If you try to just express lysozyme and holin, you get an unstable cell. Even low-level leaky expression of these genes is sufficient to cause instability and slow growth. However, constitutive expression of antiholin will buffer out low levels of holin production and keep the system off and stable. Upon induction of holin and lysozyme, the antiholin is titrated out and the cells rapidly lyse. Many naturally occurring enzymes are capable of lysing a mammalian membrane. Most of them are able to distinguish animal membrane from bacterial membrane due to the presence of cholesterol. For our studies, we needed to identify ones that were sufficiently specific to animal membranes and would function at physiological pH. By screening 37 proteins from 11 organisms, many of these proteins turned out to be too toxic to express in the cell. However, the phospholipases and streptolysin genes turned out to be sufficiently non-toxic and functional for use. The final system works as follows. First, all bacteria encode a payload device composed of 2 fluorescent proteins. GFP is fused to a nuclear localization signal, and RFP is expressed normally. Thus, we can monitor the delivery of these proteins to watch the payload delivery process as the bacteria invade cells. The invasin gene is encoded constitutively, and if you incubate bacteria encoding just this gene with the cancer cells, the bacteria efficiently invade in excess of 90%. If you also add the self-lysis device under the vacuole-induced promoter, the bacteria will round up inside the vacuoles in the cell, indicating that they have lost their structural integrity. Finally, if you add the vacuole lysis device, the vacuoles will burst, releasing the proteins to the cytoplasm. The cytoplasmic localization is confirmed by observation of a fried egg phenotype in which the GFP is transported to the nucleus due to the nuclear localization signal, and RFP fills the entire cytoplasm.